Good afternoon, and welcome to Grand Rounds at the Clinical Center. Today we're going to have two presentations from investigators from NIMH, and they've asked me to switch the order of their talks because the first person, who is going to be Giedd, is going to talk about the normal brain, and our second speaker, Judy Rappaport's going to talk about some abnormalities. But I'm going to introduce both speakers now, and then they will sort of flow seamlessly from one talk to the other. So Dr. Giedd received his undergraduate and medical degrees from the University of North Dakota, and then he completed his psychiatry residency at the Menninger School of Psychiatry in Topeka, Kansas, and 2 additional years of residency in child and adolescent psychiatry at Duke. He joined the child psychology branch of the NIMH in 1991 as a senior staff fellow, and he's board certified in general, child and adolescent and geriatric psychiatry. So he's a child and adolescent psychiatrist and chief of the section on brain imaging in the child psychiatry branch of NIMH. His research uses cutting-edge technologies to explore the relationships between genes, brain, and behavior in healthy development and in neuropsychiatric disorders of childhood onset. Dr. Gied's research has also provided novel insights into how the brain changes in typical development, particularly during the teen years. He has received numerous honors, including the Ruane Prize for Child and Adolescent Psychiatric Research, the Blanche F. Idelson Award for Research in Child Psychiatry, and the NIH Fellows Award for Research Excellence, as well as the A.E. Bennett Award, Commendation, and the Max Hamilton Memorial Prize for Outstanding Contribution to Psychopharmacology from the Collegium Internationale Neuropsychopharmacologium. And his lecture is going to highlight neuroanatomical changes during childhood and adolescence and discuss possible implications of these changes for youth, clinicians, parents, educators, and society. And the title of his talk is going to be Child Psychiatry, New Views from Neuroimaging. Well, let me first go on to introduce Dr. Judith Rappaport, who I'm sure you all know as Judy. She received her bachelor's degree from Swarthmore College and her medical degree in 1959 from Harvard, and then she completed her clinical training at the Massachusetts Mental Health Center in Boston and at Children's Hospital in Washington, D.C. Dr. Rappaport then completed her research training in the Laboratory of Psychology at the NIMH. She's board certified in psychiatry, neurology, and child psychology. She was named chief of the child psychiatry branch in 1984, and in addition to her clinical and research roles at NIH, she is also professor of psychiatry at George Washington University School of Medicine and a clinical professor of psychology and pediatrics at Georgetown. Dr. Rappaport has concentrated her research on several aspects of child psychiatry, including the diagnosis, childhood hyperactivity, pediatric psychopharmacology, and the obsessive-compulsive disorder. And her current research has focused on the neurobiology, genetics, and treatment of childhood-onset schizophrenia. She's currently president of the American Psychopathological Association and is a member of a number of national committees and a scientific council member at the Brain and Behavioral Research Foundation, formerly named the National Association for Research in Schizophrenia and Depression. Dr. Rappaport has authored and co-authored 3 professional books and is the author of a major textbook in the diagnosis of child psychiatry, and she serves on the editorial board of numerous journals. Among her many honors are several awards, including the Charles Burlingame Award in Psychiatric Research in 2013 and the Lifetime Achievement Award from the Society of Biological Psychiatry in 2011, and the McGavin Award for Prevention of child in child psychiatry in 2002. And her talk is going to be childhood onset schizophrenia, rare but worth studying. So now let's welcome Dr. Giedd to the podium.
Thank you very much. My greatest academic fortune has been to have Dr. Rappaport as a mentor. I wouldn't still be in research without her, so it's really an honor for me to be presenting with her. For CME accreditation, the disclosure and the learning objectives. And when I tell people what I'm going to be talking about, the teen brain, I often get unsolicited feedback, such that it should be a very short talk. I mean, they found one. Isn't that a contradiction of terms? Or along this theme, it starts the elusive that my next talk will be on the Loch Ness Monster. But teens do have brains. And with the modern technologies such as MRI, we've been able to actually look under the hood at the living, growing teen brain. And if you remember nothing else from my presentation, this would be my take-home message, that not only do the teens have brains, but they're doing quite well. They're not broken, they're not defective, they're not a half-baked adult brain. They do have different features than the brain of a child or the brain of an adult, but these features have served us very well. They've really been exquisitely forged by evolution to have these properties that helped our species. So these are the big three behavioral changes that have been replicated in hundreds of studies. Adolescents have increased risk-taking, increased sensation-seeking, which may sound the same at first, but they're subtly different, and a move away from parents toward peers. And I think that the fact these are deeply rooted in our biology is supported by, it's not just human adolescents, all social mammals exhibit these three behavioral traits. The evolutionary interpretations are always speculative, but one idea is it got us out of the home, which is not a very rational move. People love us, they feed us, they protect us, they support us. Why would we leave? Why would you leave home? But it turns out that our ancestors who did it worked. It prevents inbreeding, it mixes up the DNA, creating greater variance. So I don't think it's a morally right or wrong thing. It just worked in our evolutionary history. Quite close to us at the Smithsonian, they have the Hall of Human Origins, which is a great exhibit. And one of the very small features, this is a panel on the floor in that exhibit, but I thought was actually very profound. It relates the increase in human brain size to climate change. And what the key is here is that what correlates with the increase in brain size is not the harshness of the climate, but the fluctuation itself, the change itself. And so before seeing this and talking to the curator, that, wow, times got really tough. It got so cold, only the most clever could survive. And the most clever having bigger brains and leaving offspring. But this is different than that. This is that everybody in this room has teenage ancestors whose brains are very good at adaptation. We're able to change along with the climate. And this is in contrast to our very close genetic relatives, the Neanderthals. Whose brains were actually about 13% larger than humans and survived in very harsh conditions. But it appears that Neanderthals didn't really have teenagers. I mean, they lived in the second decade in that sense, but that by the time they were 12, they already had children of their own. They can find an enormous amount from teeth. And fossils, what happens in fossils is that the air or liquid in organic tissue is replaced by groundwater that has minerals in it. The minerals precipitate out, and the minerals then preserve the morphology of that organism or structure. Well, teeth are already fossils. So our teeth are already 100% mineral. So the teeth of everybody in this room is, in a sense, already a fossil. So if you find anything, you find teeth in terms of fossil records. And in mammals, the growth of teeth correlates very highly with the duration of childhood. So while the teeth are growing across many species, that's when you have childhood. And so this is where we see that Neanderthals had this characterized in this article as surprisingly rapid growth. So if you do find a 12-year-old a tooth from a 12-year-old Neanderthal in a cave, you're more likely to find their children than their parents. So they started the reproduction quite earlier. But really what's surprising is not the Neanderthal growth, it's the human growth. We're the outliers. This bizarrely long time, we're dependent upon caregivers. No other species is even close. Even things that live longer. A very long time where we're dependent upon parents or protectors or caregivers. The longer this period, in general, the more intelligence, even in crows, in species of corvids, the more complicated the food gathering, the longer the period of childhood or dependence upon parents. But I think this was our essential trade-off. We have this bizarrely protracted period of development where our brain hasn't finished. It's just kept options open so that we're able to survive on the North Pole, on the equator, everywhere in between, even in outer space, for a while with technologies that our brains have developed. And this prolonged plasticity, I think, is really the key. And this ability to change and adapt is really being put to the test. And so sometimes called the digital revolution, digits not being our fingers, but ones and zeros. And how much of our life is spent interacting, as we are at this moment, with ones and zeros through computers, through media, through PowerPoint presentations. And revolution is often a sort of overused word, but I think when we look back in time, this will really, truly be seen as a revolution in the sense that the way that we learn, the way that we play, and the way that we interact with each other has changed more in the last 15 years than, here I have Gutenberg's popularization of the printing press, but perhaps even further. The Kaiser Foundation has been following this in longitudinal studies, and what we see is more and more of the day is spent with screen time. And so even more so now for the 2012 data, because more people have cell phones and mobile devices. In about 11 hours of the day, a lot of our waking time. And 29% of the time that we're using one device, we're using more than one. So it's really become, multitasking is the rule, not the exception. This is from a group of two twins in our study that were in our study since they were 4 or 5 years old, young, and have grown up in our study, looking at their sort of typical teen life.
Exhibit A, Rocky and Anika James, typical 16-year-olds doing all their homework all at once, and then some.
I'm working on my Spanish homework.
Chatting.
I'm working on my English homework.
Facebooking.
Trying to figure out math.
Doing Spanish.
But then I'm also doing Facebook.
Listening to some music.
And Gchat.
I'm doing 2 different Spanish homeworks.
And texting.
And I'm checking my math answers.
Scientists call them digital natives. So at ease in a world of constant connections, experts can't help but wonder, are their brains being altered along the way?
The adolescents. This is a very common concern for parents in terms of the world's changed very, very rapidly in terms of the digital technologies, and so the question comes up, is it too much in terms of how different it is from what we were doing? So this is one aspect that I've wondered about and tried to explore in terms of trying to understand these changes in the day-to-day, hour-to-hour life of adolescents. In this paper focus on adolescents, not because they're the only ones affected by the technologies, but they're young enough to embrace these changes and old enough to master the technologies. So anyone with teenagers, they're so skilled at doing these things, and the idea of a native is that they've grown up with it. It's the way that they've done things. And so I try to explore the nuances in this paper a bit in terms of learning, in terms of Google and TED Talks and the Khan Academy and this talk as well. It's amazing. Like the best minds on the planet have their content freely available to everyone, and I think it's almost magical. I think in terms of how easy we have access to information. People worry then it's too easy. Will it be too shallow? Mile wide, inch deep. Will people not do critical thinking or sustained effort? The playing in terms of video games replaced free time for many children, not only my own. And the social media in terms of Facebook is 25% of all internet traffic is social media. sites. And I think it fulfills a very basic human desire, but it's weird in terms of, as my grandma would say, just ain't natural. We evolved to interact with each other, to smell, to touch, share physical environments. So much of our brain has been dedicated to social interactions, most of the brain. That's how we survived with teamwork and getting along. That what we're doing now, we lose some of these abilities. And I'm not arguing that standpoint, but we should also point out reading's not natural either. Reading's only 5,200 years old. So for most of our existence as a species, nobody read. In terms of, so in that sense, reading's not natural either. So I don't think it's such a matter of it being natural or not. It's just, I think, goes back to this fundamental principle of we're really good at adapting. From hunting and gathering berries 10,000 years ago, living in caves, and I would think how much of our day now is spent with words and symbols and very different things. 10,000 years in evolutionary terms, the blink of an eye. So it's the same brain, but the key has been the brain's plasticity and ability to adapt. And this is really the essence of my research in terms of trying to understand this changeability in terms of how to harness it for good, how to minimize the down effects. So the way we're doing this is a very admittedly non-creative study design. We have people come here and we do a brain scan, we get their DNA and evolving kind of neuropsych battery and follow them throughout life in terms of approximately 2 intervals. About a fourth of them are healthy, happy, typically developing twins, a quarter healthy, typically developing singletons, and the other half from different clinical populations, one of which Dr. Rappaport will be talking about shortly. So to just kind of cut to the chase, the summary of what's happening in the brain in terms of anatomy, summarizing our 23 years of research, the brain doesn't mature by getting bigger and bigger. By first grade, the brain's already 92% of adult size. And so this idea of watching the brain grow bigger, initially that was what we were thinking. We'll follow these brains. At 5, they can do arithmetic. At 8, algebra. At 11, calculus or other things. We just watch their brain get bigger and their abilities get stronger. It doesn't work that way. So firstly, kind of disappointing. We'd be saying, wow, watching a 7%, 8% change over 20 years. It can be like watching grass grow or paint dry, not very dynamic. But it turns out that it's true in terms of total brain size, but the components of the brain are incredibly dynamic. And the main changes that we see are the brain becomes more connected within itself, which I'll elaborate on a bit, and more specialized based on what we ask it to do. So the connectedness part we detect from MRI through white matter changes. And the white matter is from these octopus-like cells, oligodendroglia, which just means mini branch glue, but sending out these tentacles to wrap insulating material around the axons. If you're healthy and don't have demyelinating illnesses like multiple sclerosis, your white matter goes up and up. 5th decade for women, 4th for men. So we get more and more of this insulating material, and it's a game changer. So myelinated nerves have 100 times transmission speed versus unmyelinated. So just like a copper wire insulation, the signal goes much faster. But importantly, the signal actually hops between these gaps in the myelin, the nodes of Ranvier. And that becomes very important because a great deal of all the energy in our body, 20%, is spent reloading these membranes. So when a signal is transmitted, the gates open up, the positive ions rush in, and a lot of the energy that we all use then is resetting those membranes, pumping those ions back out so they're ready to go again. The myelinated neurons, we'd only have to reload at these gaps. So the neuron is ready to fire again 30 times faster. So we have 30 times faster reloading time, 100 times speed, a 3,000-fold increase in bandwidth. So not subtle at all. I think it underlies a lot of the behaviors that we see. But it's not just a matter of pedal to the metal, the faster the better. It's all in the timing and the synchrony. So information is conveyed in the brain in the pattern of firing. That's not the side dish. That's the main course. That's how thought and emotion arise. A trade-off, though, is that as the myelin is laid down, it releases chemicals that prevent further arborization. So we have this inevitable trade-off as our brain specializes, that the connections that we use and lead to good outcomes are strengthened and reinforced, and the ones that are not used or lead to bad outcomes are eliminated. And so this trade-off between plasticity and efficiency is really what happens during much of life, particularly during the first and second decades. There are certain times when there's explosions of myelin growth in terms of ages 7 to 11 is a very dynamic time for the basal ganglia involved in movement and fine motor control. But other parts of the brain kind of have their moment in the sun, so to speak, when there's explosions of growth and pruning back. These may be related to sensitive windows that have implications for education or for illnesses. But the big message here is that the brain becomes more integrated, more connected amongst itself as it grows. And the paradigm shift in my field has been increasing use of graph theory and other ways to capture this connectedness within the brain. And this has really become more and more of the way to understand the brain imaging. It started out with, like, where's the hole in the brain, where's the damage? And then it was, okay, the brain is not damaged, but the size or shape of different structures are different in different illnesses on group average. And then it was that the trajectories of these different structures are different in illnesses. And now it's more that the relationship amongst the different structures is what changed and may change differentially in health and illness. So I'm not going to delve too deeply in Dr. Rappaport's topic, but just as an example, looking at the brains of children with schizophrenia, when we apply graph theory techniques to see how the brain is connected, we're able to discriminate better amongst the brains with schizophrenia and normal. So a metaphor would be that the way the brain works, the different parts of the brain, like letters of the alphabet. And after about 8 months, when the hippocampus finishes myelinating, what happens as we develop, we don't get more and more letters. Those letters get formed into words, the words into sentences, the sentences into paragraphs, and so forth. And that's the kind of phenomenon that we can capture using these graph theory approaches. So usually this idea of connectedness is at a millisecond or second scale. So with MEG or EEG, you look at, during a task or at rest, what parts of the brain are working together, or rising and falling together. And with fMRI, the same kind of idea, with during a task or at rest, what parts of the brain rise and fall together. But these are at scales of seconds to milliseconds, and what Armin Rosnahan and our group, the paper on, we think is very exciting for us in terms of this idea of looking not just across seconds, but across years or even decades. So the idea of what parts of the brain are growing together and developing together as another form of connectedness. And so using this methodology, what we see is a great convergence from the different methods. So when we look at gray matter changes, white matter changes, fMRI changes, they all converge upon these same connections or groups of structures that are firing together, wiring together, but also growing together. And so this gives us a whole new tool then to look at health and illness. Nancy Lee from our group this summer, and then looking at these parts of the brain, the degree to which they grow together predicts the vocabulary score. And so the kind of tightness of coupling is the measure of interest. So this is a whole new realm for us to try to look at how the brain, as it grows in health and illness, the connectivity changes. The other main change is the brain becoming more specialized, which is a gray matter phenomenon. This movie is condescending.
The brain grows like a tree. First, there is a flurry of growth. Then unused branches or pathways are pruned. And it is this pruning that gives the tree its shape for the future.
Again, not to, for this audience, but to convey that this is, by the creators of the Zitz cartoon comic strip. I don't know if people have seen this, but focuses on adolescence. But to convey directly to teens and to students in terms of that, what they do with their time and their habits and their schoolwork and sports has a big impact on how their brain is developing. It's a very empowering concept. So it's not for their teachers or their parents or for anybody else. It's that the dynamic changes in their brain are related to what they do with it. One of the messages from this has been that these changes are surprisingly enduring. And we started this study with, well, should we follow people till 16 or 18? By 16, they're already winning gold medals at the Olympics. It's only modern society that we don't treat them as adults, and those kind of messages. So we were quite surprised to see that there were some dynamic changes, well into the mid to late 20s, particularly in the prefrontal cortex, this part of the brain involved in impulse control, long-range planning, doing the hard thing, organizing. A lot of the same phenomenon that we see still at work in adolescence. But again, remember, this is as it should be. I mean, if this part of the brain was already finished and we probably wouldn't be here. And this is the two sides of this coin in terms of this very protracted type of development. So the areas of the brain that tie together information from the rest of the brain are the ones that tend to mature latest. And this can then create an imbalance between this kind of late maturing prefrontal cortex and the earlier maturing limbic system, which ignites at puberty in terms of drastic changes in dopamine and nucleus accumbens and reward circuitry. So we have a fairly long gap between these changes in the limbic circuitry and the prefrontal cortex. And this balance is what accounts for a lot of phenomena. If it was just a matter of prefrontal cortex maturing late, then we should see all these difficulties in children, not in adolescents. But this is the work of B.J. Casey, who was in our group when I first started in Dr. Rappaport's branch. But this imbalance between early maturing limbic and later maturing prefrontal accounts for a lot of the behaviors that we observe. So these changes in terms of the plasticity, they do create vulnerabilities. And that's been a big theme for us. I mean, most of the illnesses emerge during adolescence. Okay? Not a lot, or quite a few, most. In terms, not everything, not Alzheimer's, not ADHD, not autism, but most things in terms of schizophrenia, depression, anxiety, panic attacks, substance abuse, eating disorders. So this has been of interest to us in terms of, do these typical brain changes help us predict what current illness is, how to intervene, what sort of things that we could measure or track. And this has been a central theme for a group of understanding, why do things emerge in adolescence? If you're born at the risk for these illnesses, why does it wait till adolescence? It's not just stress. Childhood could be every bit as stressful as adolescence, especially if you're in a war-torn country or in poverty. So it can't just be sort of like only adolescence is stressful. I think that's one of the big sort of mysteries of neuropsychiatry, as well as the sex differences. Very, very striking. Everything that we look at as a child psychiatrist, different between boys and girls. Different ages of onset, different symptomatology, different prevalences. Things that are more common and early in life are more common in boys. Autism, ADHD, dyslexia. Things that are more common in women are more common after puberty. Depression, anxiety, panic attacks. So there's clues there. I mean, that I think haven't been explored as much. So usually we hear these like planetary type of differences between males and females. We think it's more subtle. Even people from North Dakota don't get this, but the point is that it's there are differences, but they're not planetary. They're subtle, but that these subtle differences are important because these illnesses are very different in development. And so one way to study these impacts is to look at people that have extra X or Y chromosomes. So they have 300 such people. Happens about 1 in 400 live births. People have something other than XX or XY. So we can look at dosing effects of this. So Armin and our group has been doing a lot of work looking at the kind of dosage effects of extra X or Y chromosomes. Or people with anomalous hormone levels to try to get a bit of a handle on this. And a lot of what we see as adolescents work isn't exactly an illness in terms of like there's probably not something broken, but it's still very real. It impacts lives greatly. Unplanned pregnancies, sexually transmitted diseases, car accidents, the #1 cause of death for teenage girls, criminality and incarceration. All these things have huge lifelong impacts, but they're not illnesses, but I think they still fall in the domain of adolescent brain development. So it's often, is the glass half empty or half full, and for the teenagers, not only is the glass half empty, the water's dirty, the glass is cracked, and it's your fault kind of thing, this very sort of negative, impression of adolescence. But it doesn't need to be that way in terms of the adolescence. It's actually a time of great opportunity. Our interventions should have a greater impact if we can catch things early in the course of illness. That this plasticity, is a great upside. So most teens do well. They get through this time, become functioning adults in their society. But because of this, dynamic changes, we can do better. We can, if we understand the biology and the mechanisms of these brain changes, then we can do something about it. That's different talk. Sorry. But so I'm gonna conclude there and transition to Dr. Rappaport's, talk, looking more, at this vulnerability aspect. But thanks for your attention.
Jay, and thank you very much for the, oh, wait a second, that's you. I'm giving your talk again. Where is mine? All right, it is a great pleasure to be here, and I want to thank John for inviting us here, and particularly thank the NIH for the ability to do the unique things that you really can do here. Jay had an example of something you would never get a grant for. He started with time on an MRI machine, and we figured every 2 years was about right, and when we started those studies, the gold standard was hand outlining of every structure. And we figured they'd get better programs. They've got to. And we figured 2 years was reasonable because we were yoking controls to patients and so on. There isn't anywhere else you could have done that. And it worked out very well, and the machines turned out not to be lemons and so on. But this is a very special place to work. And I'm going to talk about a rare form of a not rare disorder. And we've had the luxury to not only recruit nationally for this very severe and devastating disorder, but also to follow them longitudinally. And that gives you very unique insights with respect to development. And no disclosures, unfortunately. And what I want to cover is that some sense of recognizing these very rare and very ill patients. That turns out to be an important thing to be able to do because rare disorders are very often misdiagnosed, and in this case probably even overdiagnosed. And then to talk about the clinical and neurobiological risks that are particularly salient, and then talk a little more about a particularly interesting finding of ours to do with copy number variants and this particularly severe developmental disorder. There have been a number of older epidemiologic studies or just reports of hospitalization for schizophrenia, and this is from the UK, but they're very similar ones from France and Germany. And everybody knows about the early 20s with the sex difference with a male preponderance, but somewhat counterintuitively, because child psychiatrists often, when they're young, treat more male than female patients, but counterintuitively, when you hit psychosis, that sex difference seems to disappear. So that's not going to be an obvious one, and the question is, why should these cases happen that don't have them, and what kind of insight can that give us? So medicine is not physics. You tend to have ands in finding rather than ors. And every one of these ovals have to do with findings with respect to schizophrenia in general that have been replicated and shown to be related to have etiologic significance for schizophrenia. But the sentences and the letters that are in red are ones which is what we were after, which is where are the hints of greater salience, because early onset populations may be more biologically homogeneous or may have certain factors that leap out at you. The main point I want to make in terms of the clinical work is just that these patients, as is the case across much of medicine, early onset cases really resemble poor outcome, very severe adult patients. And one of the things we were very surprised at is we have really the luxury to spend 2 days screening, and we've been sent or called about several thousand patients. But when we've in person carried out screenings far more in depth than you would have in a typical clinic setting or clinical practice, what we found is that only about half of the patients at the end of these 2 days would be considered to have schizophrenia. And that's, I think, because child mental health caregivers so rarely see these patients that they tend to mistake, which is very understandable, a psychotic symptom, which is not rare and cuts across many disorders, for the full diagnosis of the disorder. Even more surprising to us was that we then have the luxury of having the patients who we presume to have the diagnosis spend several months, if necessary, in inpatient observation. And even more unique, used to be the gold standard of care, but you can't do that anymore, includes several weeks drug-free observation. Very unfortunate that the finance of healthcare don't let that happen much anymore. But even there, we found that once you really could observe with a very skilled nursing staff and the luxury of time, that there are other forms of disorders with psychosis. 25% of bipolar disorder, for example, has a psychotic form, that psychotic depression in children is not rare. And the biggest thing that I learned, and important cutting across all of our findings, is that anything that gets terribly severe can look like psychosis, whether it's an extraordinarily severe form of anxiety disorder, depression, obsessive-compulsive disorder. When it's so extreme that it colors every aspect of thought and life, it is surprisingly easy, even for very excellent clinicians, to not be sure whether it's schizophrenia or not. So this is not a readable slide, but the point that I wanted to make is that while most of our age of onset, and we define schizophrenia the same way as you would define adult schizophrenia, which is a mixture of positive symptoms such as delusions and hallucinations, negative symptoms, which is a withdrawal of interest and motivation from life in general, deterioration, a chronic course, and so on. But we defined our population as having to have a documented onset before age 13.